Hi everyone, my name is Jessica Guo, and I'm presenting my research on piperlongamine and piperlongamine mitigating rotenone-induced neurotoxicity and the implications for a novel Parkinson's disease treatment. To begin, Parkinson's disease is a common neurodegenerative disease that affects over 10 million people worldwide. PD is a progressive disease that entails motor symptoms such as tremor, bradykinesia, muscle stiffness, and walking and balance problems, as well as non-motor symptoms such as mood disorders, and perhaps one of the most notable symptoms is cognitive decline and the memory loss associated with it. PD is characterized by dopaminergic cell death, a loss of dendritic spines, apoptosis, necrosis, and ROS, or reactive oxygen species generation. While causes remain ambiguous for these characteristics, triggers include alpha-synuclein aggregates, inflammation, and mitochondrial dysfunction, especially complex 1 inhibition. Although these hallmarks have been extensively studied, their etiology still remains ambiguous. Current treatments, which include pharmacologic treatments such as levodopa or neurosurgical methods such as deep brain stimulation, only alleviate symptoms with none that directly address its causative pathophysiology. Thus, there exists an urgent need for improved PD therapies. For this project, because PD's pathophysiology is multifaceted, it can be difficult to find an adequate model to experiment on. However, SKN-SH cells treated with rotenone can be used as a PD model because it recapitulates PD pathogenesis. This project focused on whether two natural antioxidants, piperlongamine and piperlongamine, can mitigate rotenone-induced toxicity and its implications for a Parkinson's disease treatment. My goal is to investigate the protective abilities of these carefully selected chemicals, piperlongamine or PL and piperlongamine or PM, on an in vitro PD model. The protective effects will be assessed in terms of cell proliferation, cytotoxicity, deformation, caspase activities, and alpha-synuclein. In silico experiments will also be run to determine molecular mechanisms behind the chemical's impact. My hypothesis is that piperlongamine and piperlongamine can mitigate rotenone-induced toxicity by increasing cell proliferation and suppressing rotenone-induced apoptosis, necrosis, deformation, and alpha-synuclein through the TLR2, APO1, and MC1 receptors. In this project, for cell culture, PL, PM, and R were dissolved in DMSO and glutathione in water before all were diluted in MEM to concentrations of either 1 micromolar or 0.1 micromolar. Human neuroblastoma SKN-SH cells treated with rotenone were used to create a neuronal Parkinson's model. And cells were conventionally cultured and maintained at 37 degrees Celsius in a humidified atmosphere of 95% sterilized air and 5% CO2. The control group were wells of just cells. The experimental group were wells with cells, rotenone, and the chemical. This chemical included either PL or PM, and glutathione is a chemical that has been well established to mitigate Parkinson's disease, so that was used as a positive control. MTT assays were conducted to measure cell proliferation, and LDH assay to measure cell cytotoxicity, computer-assisted cell image analysis to measure cell deformation, caspase 3, 7, and 10 assays to measure apoptosis, alpha-synuclein ELISA to measure alpha-synuclein levels, and molecular docking to determine molecular mechanisms behind the chemical's impact. For the MTT cell proliferation assay, a 96-well plate was pre-treated with PL and/or PM, and steps in the Cayman chemical assay kit were followed. Absorbance was then measured at 595 nanometers using an iMark microplate absorbance reader. And then for the LDH cell cytotoxicity assay, it's the same cell culture conditions as the MTT assay, and steps in the Invitrogen Thermo Fisher kit were followed. Absorbance was then measured at 490 nanometers. For the computer-assisted cell analysis, cells in a 6-well plate were stained, and then steps in the HEMA3 staining kit from Fisher Healthcare were followed. Images were then taken under a binocular microscope, and image J analysis was conducted to demonstrate differing effects of each chemical at a histological scale. For the caspase 3, 7, and 10 assay, a 6-well plate was pre-treated, and then steps in the caspase assay kit from G Biosciences were followed. Absorbance was then measured at 415 nanometers. For the alpha-synuclein assay, it's the same cell culture conditions as an MTT assay, and steps in the ELISA kit from Boaster Biological Technology kit were followed before absorbance was measured at 415 nanometers. For molecular docking, the structure of the ligands piperlongamine and piperlongamine were acquired from the PubChem database. The 3D receptor structure was retrieved from the protein databank. Pyrex was employed for analysis, and the Biovia Discovery Studio Visualizer was used for more precise 3D visualization of the ligand receptor binding. Significance results for all of these tests were analyzed using a one-way ANOVA and then a post-hoc Sheff. Results with a p-value of less than 0.05 are considered statistically significant. In the MTT cell proliferation assay, rotenone caused cell death, whereas PL, PM, and the group with both PL and PM demonstrated neuroprotection. PL and PM together demonstrated the greatest attenuation as it increased cell proliferation 39% in comparison to R, which suggests that, in the short term at least, PL and PM together is the most effective at mitigating cell death in PD. Looking at these pictures, when you compare the control group cells to cells treated with rotenone for 24 hours, the number of cells per unit is significantly less, while on the other hand, pretreatment with PL, PM, or both revives the number of cells. LDH levels indicate cell membrane damage and necrosis. When the cell membrane is compromised in any way, LDH, which is a soluble yet stable enzyme found in every living cell, is released into the surrounding extracellular space. The presence of this enzyme in culture medium can be used as a marker for necrosis. Necrosis is cell death that is characterized by the swelling and rupture of intracellular organelles, which eventually leads to the breakdown of the cell membrane. In this experiment, rotenone significantly increased LDH release, indicating an induction of necrosis, whereas all other chemicals decreased cell cytotoxicity, therefore decreasing cell necrosis caused by rotenone. Dendrites receive and transmit information between neurons, and they play a critical role in memory retention. Reduction in synapse number and dendritic size is connected with neurodegenerative diseases such as PD and are believed to be a major contributor to altered and impaired cognition. Dendritic length was measured, and as you can tell, compared to the control, cells treated with R expressed a significant decrease in dendritic length. PL displayed the most significant attenuation of rotenone's harm, but PM and the combination treatment of PL and PM also displayed significant increases. As you see in this figure, the control has many long dendrites between cells, whereas rotenone does not. Compared to rotenone alone, pretreatment with PL and or PM significantly increased both the length and the number of dendrites. PD cells also usually have rounder cell bodies. Increased roundness indicates an inactivation of the cells, which means that the cell is dying. Compared to the control, R significantly increased the roundness of cells, and PL and or PM significantly decreased roundness when compared to rotenone alone. Rounder bodies correlate with greater mitochondrial dysfunction, so it's possible PL and PM are able to directly target the mitochondrial inhibition present in PD, and these results indicate that pretreatment with PL and or PM was able to prevent neurodegeneration through the modulation of cell morphology. As you can see, rotenone-treated cells are much rounder than the other treatments. Also, the number of rounded cells for rotenone is much more than the other treatments. Caspases are cysteine proteases with a well-defined role in apoptosis and inflammation. Rotenone significantly increased apoptosis by upregulating caspases. PM was able to completely block rotenone's impact on apoptosis by downregulating caspase activity, and while to a less great extent, PL also significantly mitigated rotenone's effect on apoptosis. This proves that through caspase downregulation, the treatments were able to attenuate rotenone-induced apoptosis. Additionally, the cleavage of caspases 3 and 7 are triggered by the stimulation of the intrinsic mitochondrial apoptotic pathway by ROS, indicating that ROS levels were attenuated by the treatments. Alpha-synuclein accumulation and aggregation is a well-known hallmark of PD, but the peptide remains a difficult therapeutic target because of its multiple distinct roles that are relatively unstudied. In this experiment, R decreased phosphoserate 129 compared to the control, while both individual and combination treatments of PL and PM increased levels compared to the control and rotenone. Through previously run tests such as the MTT, LDH, and caspase assay, it was concluded that the target chemicals were protective. Initially, these results contradicted all of those tests. However, it was later discovered that this experiment was one of the first to expose the bilateral abilities of alpha-synuclein. Endogenous monomeric alpha-synuclein has a neuroprotective role, whereas the conversion to form insoluble fibril aggregates is responsible for neural dysfunction. The fibrils should be targeted, as targeting the monomeric forms instead consequently inhibits the protection of normal alpha-synuclein. Thus, this definitively provides a new therapeutic target to treat alpha-synuclein aggregates instead of its favorable form in an effort to mitigate Parkinson's disease. Molecular docking was conducted to predict binding affinity, which is a measure of how well a drug binds to a macromolecule. A binding affinity of approximately -5 kcal per mole or lower is usually considered strong. The APO1/CD95 or FAST receptor is important for apoptosis regulation, the toll-like receptor 2 or TLR2 for alpha-synuclein management. Alpha-synuclein is a hallmark of PD, and mitochondrial complex 1 or MC1 inhibition leads to increased cell death in PD. PL and PM both had strong binding affinities for each of the 4 receptors. PL had a stronger binding affinity for TLR2, and PM had a stronger binding affinity for FAST, alpha-synuclein, and MC1. This is a 3D visualization of ligand receptor binding site, and as you can see in the photos, PL and PM are able to bind to TLR2 and MC1 well. This figure shows the pathogenesis of Parkinson's disease as well as what contributes to the increased depth of dopaminergic neurons in PD. The FAST receptor is a death receptor that triggers apoptosis. So because PL and PM had high binding affinities for FAST, and FAST activates caspases 3, 7, 8, and 10, which mediate the mitochondrial apoptotic pathway, PL and PM are possibly able to directly target the FAST receptor to downregulate caspase 3, 7, 8, and 10 and protect the mitochondria. Results from the in vitro caspase assay also align with these results. The TLR2 signaling pathway contributes to alpha-synuclein pathology and neuroinflammation. So because PL and PM exhibited strong binding affinities for TLR2, this suggests that the 2 target chemicals can directly mitigate alpha-synuclein and neuroinflammation through the TLR2 pathway. It's also widely accepted that alpha-synuclein is a prominent hallmark of PD. The in silico and in vitro results show that PL and PM are able to target alpha-synuclein, lending implications that the chemicals can attenuate PD by preventing the harmful accumulation of this protein. MC1 dysfunction translates to elevated ROS levels, which leads to alpha-synuclein accumulation and increased neuronal cell death. Docking experiments predicted high binding affinities to MC1 for both PL and PM, which suggests that the 2 chemicals can mitigate rotenone or PD's harm through complex 1 protection. Thus, PL and PM are able to counter the death of dopaminergic neurons by modulating neuroinflammation, preventing ROS and alpha-synuclein accumulation, as well as downregulating caspase activity. In conclusion, 2 highly accessible alkaloids, PL and PM, can provide strong protection against rotenone-induced neurodegeneration. Each chemical exerts significant protection on neuronal cells at cellular and molecular levels. The bilateral nature of alpha-synuclein was also exposed. PL and PM can directly target multiple PD pathways to mitigate disease progression. So overall, piperlongamine and piperlongamenine have the potential to be novel treatments against Parkinson's disease and slow disease progression. In the future, studies directly measuring ROS, complex 1 activity, and neuroinflammation should be conducted. In this project, only 24-hour treatments were used, so in the future, there should be long-term studies done. In vivo models should also be tested on, as certain markers such as the gut-brain interaction as well as the blood-brain barrier cannot be measured with an in vitro or in silico model. Here are my references, and thank you so much for your time today.